I want to officially welcome you all to our first inaugural Patty Marshall Lecture on Genomics and Equity. It's amazing to see so many colleagues and friends here, both from our local institutions as well as national and international guests. As many of you know, Case Western and the Department of Bioethics has had a long history of scholarship, teaching, and service related to the ethical, legal, and social issues surrounding advancements in genetics and genome sciences. In 2004, Case was awarded one of the first centers of excellence in LC research by NHGRI and established the Center for Genetic Research, Ethics, and Law, known as CWRU, first under the leadership and vision of Eric Youngst, who's here in the audience, thank you, yay, yay. And then led by Richard Sharp from the Cleveland Clinic and Patricia Marshall. Addressing equity implications of advances in genomics has always been one of the central missions of CWRU, and as genomics becomes more integrated into clinical and public health settings, we believe it is essential to be more explicitly focused on efforts to work towards addressing the potential implications that genomic information could have for our understanding of health disparities and making sure that we assure access to genetic technologies. In this spirit, today we are establishing a new center that will carry on the legacy that Eric, Rich, and Patty have laid out for us by launching the Bioethics Center for Community Health and Genomic Equity, otherwise known as C-Change. We believe, we believe that the goals of this new center not only advance the previous work of CWRU, but are supportive of the ongoing equity-focused work in the Department of Bioethics, led by Mark Alicio, and by the equity and community-centered goals laid out by Dean Gerson's vision and strategic plans for the School of Medicine. Thank you to those in the audience who have already signed up to be affiliates of our center, and we're looking forward to having more of you involved. There are flyers right outside if you're interested in becoming involved with a QR code that you can link in and become an affiliate of the center and get to know more about our future activities. But now on to our honoree. Similar to CWRU, equity and social justice has always been an essential and integrated part of who Patty Marshall is as a scholar, as a teacher, as a mentor, and as a human being. Patty retired this year after over 20 years of dedicated service to our department and the School of Medicine, and we could not think of a more fitting tribute to Patty's own legacy than to establish this annual lectureship in her name and to kick off our center. One of Patty's mantras, whether in her work with underserved communities locally or her work in Nigeria or other international settings, has always been to ask, who's at the table? And for genetics and genomics to truly address issues of equity and community health, we must make sure that community voices are essential driving forces shaping the future of genomics in healthcare and public health settings. As a former student and now colleague and friend, it is such an honor to be able to establish this lectureship in her name. Before handing the mic over to introduce our esteemed speaker, and let me just say how lucky we are to have Dr. Charles Rotimi here to be our first inaugural lecturer. I wanted to thank just a few people. I want to first thank Dr. Marcia Mickey, who's Associate Director of our new center, for her invaluable work and energy in putting together not only this event, but the center. I want to thank the Department, Mark and the Department of Bioethics for all of their support, including all of the staff support in making sure that today happens. And I want to give a very special thanks to someone who, without whose tireless work and dedication to our department and the center, these events could not happen. Roselle Ponceron, we have a little.
Roselle, thank you so much.
And before handing the mic over, we have some small tokens of appreciation and love for Patty and for Charles, to thank them for being part of our center launch. Here's yours. Thank you so much. And Charles, this is for you.
Thank you.
Thank you so much for being part of today's lecture and for being part of our center.
Thank you.
Wow.
So
Awesome.
With that, I will hand it over to our esteemed colleague and friend, Patty Marshall.
Get out of here. I feel totally free saying that to him because he's just a dear friend. And I trained him and Shlomi along with Eric, We did a good job, Aaron. I've been told, can you all hear me if I go a little bit like this? Can you hear me? Because I plan to be dancing on the stage. I've been told to keep my remarks short. That can sometimes be difficult for me because I am filled with stories. But I am going to keep my remarks short today. Charles, my colleague and dear friend, for how long? 1,000,000 years.
+1.
We've traveled the world together. So a few things that you can see from here. NHGRI, he's the scientific director of intramural research. NHGRI, that acronym stands for National Human Genome Research Institute at NIH. And he's also a distinguished investigator. Well, that's pretty cool, Charles.
Very cool.
He has so many accolades. I could spend so much time just talking about them and him as both a colleague and friend. But I'm going to let that go because you all have his bio, and if you look him up online, you're going to be inundated. You know, you're going to need a glass of wine or a little bourbon or a good strong beer.
All three.
One after the other. What do you start with? Also, I want to make this one remark. As you know, Charles is a leader in the in international research on genomics, the globalization of genomics, particularly on the continent of Africa. So I'm going to put your accolades here, okay? They're going to be over here, Charles, but not forgotten. You know, and I'm just saying, thank God you've got people like me and your friends to keep you anchored on the ground, you know? So I want to focus on Charles as a character, as a man, as my friend, as my collaborator, as my colleague for so long. And I want to start, oh, I don't like that light. I want to start by, I want to start with a poem written in 1987 by a Nigerian poet, and it goes like this. Rise, take your dream to the end of the street. I'm start to cry. Then stretch the street and take it to the end of your dreams. Do you remember this, Charles, that poem? And this so very much, for me, embodies the spirit of you, of who you are. So, you know, you might think, well, how have you walked down this street that has stretched and stretched beyond? And I want to say, you have walked with integrity and respect, enabled by the transformative power of imagination. So what I want you all to think about is this notion of imagination, the transformative power of imagination. What do you see? What you see is the beginning of a thought, translated, shifting a perspective, moving forward, changing a bit, and moving again, moving on. That is how you have walked that street, stretching it to enable your dreams. And I have so many stories, I can't go there. But one short story that speaks to your respect and integrity. Charles and I have done work together for a long time. And I forget what year this was. We were at Loyola then, but I went to Lagos with you. I was doing some consultation work for the for NBAC, the Bioethics Advisory Committee for the government. Anyway, so you were meeting with a team in Lagos, your team. They were out in the field, and of course, Dr. Rotimi here he comes. He's going to speak some wisdom, and you certainly did. The beauty of this goes to two issues. One, they felt comfortable asking you a question that was kind of sensitive. Their job was to be in the field collecting DNA samples from a trio of child biologically related mother, father, okay? But his question was, what do you do if you walk into a homestead, a household where different families are living, and there's another wife who says, well, I want to join this study too. I want to donate some of my DNA for your study. And without hesitation, the subtext, I'm thinking, no problem. What you said was, not a worry. Invite her to join the study. Let her donate a sample of her DNA. Didn't have to use it for your particular study, but why create a problem for the family? Let the narrative of this family be in peace. One example. Another example it has to do with H3 Africa. What does H3 Africa mean? Human Heredity and Health in Africa, an initiative to build the capacity for genomic research on the continent of Africa. No small vision. Where did it start? In the head of this man, right? And how many nights in Nigeria, in particular, in Ibadan or Kenya or Uganda, other places, South Africa, thinking about how to do this. Remember the transformative power of imagination. Thinking about how to do this. Drinking cognac with it always helps the imagination. Good glass of Hennessy. Okay, that idea came into formation, shaped, reshaped, transcended, and grew, and is now a hugely successful initiative. You were instrumental in that. I've been involved for many years I was a founding member too. All right, I'm just saying. But that speaks to, those are just two small stories. Oh, Charles, the stories I could tell. As you know, be grateful that I'm not going in the direction of those other stories.
Yeah, I was ready to hide under this.
It's like, wait, Patty, stop. But I want to say one more thing to you personally, and then I'm going to go down here so I can see your slides. So, Charles, my dearest friend, I celebrate the story that is yours and what it will become. There's so much room on that street towards dreams. Thank you. I honor you.
Thanks, and thanks for coming for this inaugural talk. And as you heard from Patty, I'm not going to say too much about it. You know, we've been friends for so long and dear friends. And yes, we do have stories that can be good, you know, bad and ugly. But it's been many years. In fact, when I was flying down last in the afternoon yesterday, on the plane, I was counting, as you know, just the number of countries I have visited with Patty, and I stopped at 9. So you can see that we really have had a very long history, all the way from Loyola before she came to Case Western, and then I moved to Howard, and then NIH. But the first thing I wanted to really share with you is when Patty was saying humble background, I really, my story is really a humble one. And this is why I do care quite a bit as to who's on the table, you know, when we are making decisions and we are talking. And so that's my, you know, father and my mother. My mother did not have any formal education. My father was a school teacher. And that city there is Benin City where I, you know, indeed grew up. This house is still there, and I still enjoy it whenever I visit Nigeria. The other thing I wanted to share with you is last night I was joking at the table when we were having dinner that this was the first paper I published in my life after my BS degree in the University of Benin, where I was studying aflatoxin, the effect of aflatoxin, that is a hepatotoxin, in Nigerian beverages. And palm wine, I don't know if anybody here has had the opportunity to taste palm wine. It's actually quite sweet and very nice, especially when you get it the days coming down from the palm tree. After that, it can ferment and the alcohol content increases. But the research I was doing here was really to understand how this Aspergillus flavus, a fungi that grows on this beverage, produces aflatoxin, which is, again, very toxic to the liver. But the point I want to make here is that I was doing health disparity research, and I didn't know it, because who doesn't have access to refrigeration? Again, these are people who are of less income and things like that. So, but shortly after that, I came to America. And this lady here, my mom, really surprised me, and I'll never forget that surprise. I applied, you know, to 2 schools, one in the UK, University of Manchester, to read petrochemical engineering, and then to University of Mississippi, Ole Miss, to continue my biochemistry. And I was accepted to both, but Manchester was too expensive, so that was out of the question. So I went to talk to my parents, and I said, please, I would like to go to this school, and but I don't know if we have that kind of money. It was $7,000, you know, to enroll. And to my amazement, this lady said, "Oh, I can pay for 1 year." And I don't know where she got that money from. And she said, "My son," this is exactly what she said. She said, "My son, this is what I've been saving for all my life." And that is how I came to America. And I was guaranteed 1 year of school fees. And after that, I was, you know, again, on my own. But what it speaks to is how your parents and that guidance really can nurture you and give you possibilities. And I really cherish that. And Patty got to meet her. I have some pictures later on that you'll see that, again, sort of solidifies our relationship and friendship over the years. Now, the story I want to tell today about disparity, inequity when it comes to genomics really starts from the very beginning. I want us to, with the successful sequencing of the human genome. The way I look at our genome, I look at it as a history book. And that history book, we are just beginning to learn how to read it. And it has unbiasedly captured where our ancestors have lived, all the things that they have to have survived, in terms of toxins, infections, and things like that. And we are just beginning to read this book. And the question is, what is this book telling us? Again, the first sort of publication was 2001, and then recently, the 8% gap that was left, we filled it up, and with using new technology. So now, all of these stories, all the things that we're doing in genomics continue to be consistent about one major fact, and that is we are all Africans beneath our skin. If you go far back enough in the trace of your ancestry, you will end up in the geographical location called Africa today, because that is where humanity started from about 300,000 years ago. And we spent most of our evolutionary experience so far on that continent or that geographical area. Before, again, after the split from chimpanzees, So again, that is really what one of the key messages I try to give in this kind of talk. So, but of course, we know we like to travel. We know to go to different places. So some of us ventured out about 100,000 years ago and populated the rest of the world. It is because of this common history that we do share considerable amount of our genome. I've talked to some genomicists who actually do a lot of sequencing. They said the similarity in the genomes of different people is so much that it's almost boring. And that is really something that we need to, again, fully appreciate. So, you see, that small number of people that left Africa, about 100,000 individuals, 100,000 years ago that left Africa, only took a subset of the human variation that existed on the continent at that point. This is what I call the original bottleneck. And they only took a subset. So that's why we share a lot of, old things in our genome, but there's a subset of our variation, of human variation, that you can only study in the continent or the place we call Africa today, because they were not carried out. Although as humans continue to evolve, continue to travel, we carry some of those, but majority of it is still residing on the continent of Africa. And that is why I always say, if we are so similar, why are we different? So we see the original reason why there are differences across the genome, and the forces that have influenced our genome is not uniformly distributed, and therefore the variation that those forces created are also not uniformly distributed. It's not human variation is not random. There's a structure around it, So, and again, like I said, in terms of adaptation to different environment and things like that. Now, HapMap, this is really where Patty and myself really started working together. We were the PIs that were funded to engage 3 African communities for the International HapMap project. The goal of HapMap was really to help us to understand the scope of the variation that exists. The human genome told us the address of where these things are, but we needed to understand, when you take me and you take Patty or you take somebody else, how do they vary? What are the areas that we see variation? And that's what HapMap really did. And we engaged the Yoruba community in Ibadan, Nigeria, and the Luhya in Kenya, and also Maasai in Kenya. Again, these are photographs of us doing different things. The top one there, I think you see Patty at the end of that. That's where we were doing community engagement in Ibadan, Nigeria, and this is in Kenya. In all of this, I was standing on the shoulders of Patty and also Shamir Roya in terms of the ethical implications of the things that we are doing. And I want to say that of all the genomic projects I've been engaged in, there's no one that have even come close to what HapMap did in terms of community engagement. And it was expensive, but it was thorough, and up to today, we still send information to these communities about how their samples and resources are used in genomic science. So it's been, again, quite robust. So the HapMap also allows us to understand how things travel together so that we are able to summarize variation and then put them on an array and do genome-wide associations, that is the workhorse for us today in terms of complex disease research and trying to understand the underpinning of genetics in complex disease research. And again, some of those samples went to become part of the 1,000 genome. But the point I want to make with this slide is really to show, again, that the variation we see and the distribution of variation, again, can structure in terms of ancestry, in terms of geography, okay? Again, with African ancestry individuals displaying the highest number of variation, whether it's single nucleotide polymorphism or non-synonymous or loss of function variants, you always have more of those. And the way I try to summarize this, for example, I always challenge myself when I'm communicating. Can I explain this to my mother? I always challenge myself about that. So the way I try to express the variation that we see, especially in African ancestry individuals, because it's so huge, is if I take this Yoruba person and the Maasai and I compare the two of them, they are more likely to be different from each other than when I take one of them and compare them to this guy, such guy. That is the diversity that you see across African populations, And that is why I also make the statement sometimes that the way we fund science is not a rational process either. And that is, if we were sequencing the human genome and we truly wanted the biggest bang for our money, given limited resources, all you need to do is just treat that somewhere in the continent of Africa, especially sub-Saharan Africa. And anybody you sequence there, probably going to give you more information than from any other part of the world. So that's weird. Now, the other way I also try to explain this variation, diversity that I see across African population, why I continue to say the way we label ourselves, the way we call ourselves, our identity, and things like that, all are very, very important. Now, this is a paper I wrote with Lynn George, some years ago, where we actually showed very clearly that when we use labels like African or Black or white or European, Asian, we can distort our understanding of how things vary across the genome. This is a very good example of that. Abacavir is a drug for treating HIV, and it can result in hypersensitivity. And there's a particular HLA that can help us identify individuals who can have this hypersensitivity. The frequency of that HLA is almost 14% in Maasai and 0, in the Yoruba. And African Americans about 1%. So if you use the label Africans or Black, you can see how you can radically distort our understanding of variation, and you can arrive at a very wrong public health decision in terms of who to screen. Again, the issue of diversity. Now I really want to talk about the title of my talk, and that is that lack of diversity is an ongoing challenge in genomics, and also the fact that variation is not random. So engaging different world population in genomics, making sure that we all are participating. I look at it from a scientific imperative point of view, and what do I mean by that? I say it's a missed opportunity to understand history and biology. This is an example. PCSK9 and LDL is our understanding and identification of that drug that we use today to treat so-called bad cholesterol. In all human populations, we were able to make that discovery by focusing on African Americans because the prevalence of the variant is about 2% and very, very rare in European populations. So we were able to capitalize on that information to make this discovery. But what is important in this message is the fact that we discovered it in African American doesn't mean it's not going to work for other world population. All we did was to take advantage of where we see it more and then study that population. And again, increasing the diversity. Now, in terms of social justice, recently, there was some discussion about some African American or people who call Black were at risk of elevated risk of heart disease. And it turned out that that conclusion was reached in a very wrong way because the reference database that was used did not have enough representation of individuals called African American or Blacks. And that resulted in a big problem. You were calling things pathogenic when they were indeed not pathogenic because you just did not represent it enough. And I'll show you further examples of that. And it brought me to ask questions like, will tomorrow's medicine work for all? Are we going to truly engage, use genomics to develop drug therapeutics? And if we do, will those therapeutics be applicable to all human populations? So what do I mean by lack of diversity? I have 3 cartoons here that I'm going to use to explain it. This is, if you look at today, all of the things we do in terms of genome-wide association study or trying to understand complex disease research, you see the African population is almost non-existent. Okay, that's one way of looking at the diversity. Another way to look at it is look at it in all world populations. And you will see that majority of what we have done is in European ancestry individuals, are very limited in Asia, again, almost no existence in African and other, world populations. And this, again, sort of say it in a different way. And that is, if you put where we do work compared to the populations of these various areas in the world, you will see there's also an imbalance between the actual population and where we are doing most of this work. Again, this is not to blame. It's just to highlight the fact that this is what has happened the way we have funded science. And also the fact that when we were getting the gains of genomics, we were concentrating on areas where we already have samples. And we continue to emphasize that. And that's continuing to set us back, as we move forward. So why is it important to have diversity in genomics? And why is it a scientific imperative? This is a paper, that I published from my lab. But I want to, first of all, bring attention to the fact that we have been extremely successful in identifying genetic basis of type 2 diabetes, especially in European ancestry individuals. Over 400 locations in our genome have been associated with it. Despite that success story, in our study that we've been doing in Nigeria and Kenya, we have indeed been able to make novel discoveries. Okay, so this variant there called ZRMB3, we looked at it in available public databases. It's very rare in the world population, but it's relatively common, close to 2% in African populations. Now, you would ask, why is it that that variant was not discovered in samples almost 1 million European ancestry individuals? Again, it's because of that rareness, in those populations. And therefore, if we have not studied African populations, we probably would never make this novel discovery. And I want to show how important this discovery is. Because we did not know what ZRMB does, we actually went to model organisms, zebrafish, and we looked at the consequence. If we silence or knock out ZRMB3, what happens? You see this massive reduction in beta cells, which of course we know will likely have implication for insulin secretion and therefore type 2 diabetes. And it turned out to be quite correct. So where we silence ZRMB3, you will see like right here, those cells, when we challenge them in a high glucose environment, they are unable to respond adequately in terms of insulin production. So again, we are following this more and more, in my lab. to see how we can, again, take this maybe to therapeutics or at a minimum, use it in terms of clinical practice. This is another example illustrating the importance of diversity. This is a charge consortium, very large collection of samples, over 600,000. But this particular study, led by Amy Bentley in my lab, is for about 400,000 individuals. Now, 9 out of the 13 novel variants that we were able to discover in the context of gene-environment interaction, 9 of them were from African ancestry individuals, or trans ancestry that included African, individuals. Now, this was not always due to allele frequency. So you see all of the African ancestry samples, there was some association, but when you look at European, Asian, and Hispanic, there's really no association whatsoever. And why was this discovery important? It turned out that this discovery, this particular SNP here, is important in terms of taste receptors. And I was absolutely surprised when I started looking in the literature that I noticed this thing. And that is, for whatever reason, African Americans who smoke prefer menthol cigarettes. And close to 80% of African Americans who smoke menthol cigarettes. And I would say, why? Why is that? And it turned out that the consequence of smoking menthol cigarette can be actually quite severe. Because it reduces irritation, you actually smoke more and you inhale more. So you are more exposed to the carcinogens. And it turned out that there is a receptor, a G-coupling receptor that individuals from FDA and also NIH discovered that was actually very, very high in African Americans, and therefore begin to think about maybe there is a genetic underpinning to the preference itself, to menthol, cigarettes. So again, these are all of us, further studies. But when this study came out, this was one of the reasons that propelled FDA to want to ban menthol cigarettes. Okay, so you see how engaging different populations indeed do benefit. But cautionary, I always caution myself and my lab, and that when I say African American or African ancestry, I am not saying a genetically uniform group of people. It is critically important for us to understand that. So these 3 charts there are 3 individuals who are African Americans. You see somebody almost 90-something% African background, and the other person almost over 90% European background. And then you have somebody in the middle who sort of share half and half of this. So when you're looking at the genome of an African American, all bets are off. You really have to know where you are in the genome and where that came from. So the story here is that African American, the only way I can truly define it is the descendant of the Middle Passage. Everything else after that is really fuzzy. It's a multiple part of ancestry and multiple parts of environmental exposures and things like that. And we took that understanding to actually make some statements about lipids. Now, of all the health disparities that you hear about, triglycerides, HDL, are not one of those. African Americans tend to have much better profile when it comes to triglycerides, to the point where in my lab, when we ask, say, triglycerides in African American, I almost tell them to go repeat it because the values are so low. And I've always been very curious, why is that? So it turned out that when you plot triglyceride level or HDL level against African ancestry, in increasing proportion of African ancestry, triglycerides goes down and HDL goes up, which was telling us that this may be genetic, And so we wanted to study it because there's no reason why people who live in very radically different environment would be sharing something like that. So we sequenced several lipase genes. Just to cut the story short, it turned out that for a particular variant that was very, very important for triglycerides, LPL variant, the RS328, it turned out that depending on where in the genome the African American is, if it's under an African, if they have two copies in that background in an African, they look exactly like West Africans. So the African Americans look exactly like West Africans. If the background is zero in terms of African ancestry, they look exactly like Europeans. But these are all individuals who identify as African Americans. So you can see the importance of this kind of work. Again, to me, I really, this was a very wonderful discovery, for us, and it helps me to make the point, in terms of diversity, and making sure that we know that when we say something like white, black, also, we are really mixing genetics in a way that may not be very useful in the practice of precision medicine. Now, we also took advantage of the admixture in African American, but the point I want to make here is this in the next slides, and that is kidney disease. African American makes up about 13% of the US population, but they make up 32% of kidney failure. Now, they are 4 times more likely to end up, in terms of failure, in dialysis, and all that. Why is this? So, a group from NIH and Hopkins arrived at some kind of hypothesis because it makes sense, and that is, given that the sort of average genetic pool of African American is about 20% European and about 80% African, it would make sense that if indeed this is coming from Africa, it should track the African ancestry components. And that is what actually happened. And this discovery was made on chromosome 22. Initially it was NHGRI, but it turned out after sequencing that it was indeed the APOL1. Now, why is this story important? And why is it important for diversity? This is a highly toxic variant to your kidney. The likelihood of failure is extremely high if you carry this variant. The risk is over 10 folds. Now, it's 40% in Yoruba, 20% in African Americans, almost absent in Europeans and Asians. Now, this does not make sense evolutionarily. Things that are bad for you are supposed to be eliminated. But this rose to very high frequency. What that means is that it's actually protective against something else. And that something else kills you first before you get kidney failure. And that is trypanosomiasis infection, the so-called African sleeping sickness. Okay? And that is why it rose to such high frequency. Now, I also want to make a point here, is that now when we look at it in African Americans, or people who call blacks, in the United States at least, you will see that 1 in 5 of individuals who carry this variant will develop kidney disease. And 70% of people who are, in kidney failure are due to this particular, variant. And it is also very important in terms of kidney transplantation. Now think about it. If I carry that variant, I should be very, very careful to donate any of my kidney, okay? Because the likelihood of the one that remains to fail is very high. Now, it's also very important to the recipient of my kidney, because if they go through that operation and accept my kidney without knowing the status of the fact that I carry APOL1, you can see that they are probably also going to fail. Their kidney is going to fail. So it's already having very serious medical implications. Now, the most important point I want to make in terms of diversity and us human beings and the way we relate to each other is this map. This is the map of the world where we now have traced the APOL1 variant around the world. You will see it is everywhere now. Why? Because we human beings, one of the best things that we do in life is that we travel and we like to share DNA. And we just continue to build this complex web. So the fact that something was local doesn't mean it's going to remain local. That is not the human story. The human story is that we move and we spread things and we continue to. So when I say something is specific to an area, it's specific within a time frame. We have to bound that specificity, in terms of time frame. More importantly, there is no way to use skin color to identify who will carry this variant because of gene flow, admixture, and all of that stuff. So you see it in individuals who may phenotypically look European, because again, if you look at the ancestral background, you will see the reason why. The other reason for diversity is now we know we also share DNA with the other part of our families in terms of Neanderthal. A lot of the people that left Africa some years ago, again, also share DNA with Neanderthal. The question is, do those part of the genetics or the genome, which is about 2 to 4%, sometimes 5%, do they have any health implications? And it turned out the answer is yes. For type 2 diabetes at least. There's a variant in the solute carrier 16A11 that shows very clearly that when the variant is very important in type 2 diabetes, but when they try to track the ancestral background of this particular variant, it turned out that it was as a result of introgression from ancient genome into Native Americans or people we call, Hispanics, today. So again, showing the importance of that kind of understanding diversity. There's no way we could have understood this as clearly as we did if we were just looking at European ancestry populations or Asians only, or even Africans in this particular case. So diversity, again, important. Now, Asian DNA also played its head in terms of COVID. Okay? This study was published in the New England Journal of Medicine on chromosome 3. There was a very strong association with some very specific variants. It turned out that when the group from Germany looked at also the inheritance and where that came from, it turned out that it also came from the Neanderthal integration. Now, why is this important? It is very common in people from South Asia, about 16% in Europeans. Now, during the COVID epidemic, it was noticed in the UK that people from Bangladesh were dying more than the other background individuals. And it turned out they carry a very high proportion of this particular genetic variant, and that may be exposing them to higher mortality, in terms of death, from COVID. Again, seeing the importance of diversity and engaging the world. Now, to me, this is the most outstanding example that I can share with you that can really drive why we need to engage the world in genomics. There is a local community in Argentina that have been exposed to arsenic-contaminated water for generations. And as a result of that, they have actually developed protection. They've picked up a variant that can help them to metabolize arsenic to a less toxic, And it's so specific that neighboring communities that were not exposed to that water don't have that variant. So you can see that if we did not engage that kind of community, first of all, we will not understand that we actually have the capability to adapt to such an environmental toxin and the implication of that and what is the importance in terms of biology. Again, for me, it was really, really a wonderful example. And now I want to end talking a little bit about the work we did in terms of H3 Africa that Patty was referring to. For me, this is sort of like, when I think about H3 Africa, I think about a dream. When you dream and you say, oh, can that be real? So for me, it was really a wonderful thing that we did. It's not just Charles, it's a lot of us. But what was important here, for me, the driving force was not whether what we can gain professionally, but it's really the fact that I felt very strongly from my experience in participating in Half Marathon Genome, that if we do not engage that continent, that I have shown you the importance of in terms of genomics, that we are really going to miss out. Globally, and also the people of Africa are going to miss out. So that was really the motivating factor here. And we have H3 Africa has done so well that it's beyond my wildest imagination. The money that we brought is really small compared to what it has done in terms of changing culture. African scientists can go anywhere today in terms of genomics, and they will be respected, and they will not be questioned. When I first started, the first set of reviews I get from reviewers that, oh, you cannot do that in Africa. You cannot do this in Africa. I used to complain to Francis Collins, but we were in there, and but now you don't get such comments anymore, because we've proven over and over and over that you can do this kind of work, and we have trained a lot of people. Now, going back to the diversity that I talked about, please guide me in terms of time. I know I can keep talking.
2 or 3 more minutes.
Okay, I'll wrap up and get to the pictures that I want to use to embarrass Patty. No, I'm kidding. But again, this is another way that we can look at some of the products and benefits of engaging comprehensively something like H3 Africa. So we did some whole genome sequencing. We made discoveries of over 3 million novel variants in these 500 or so individuals that we sequenced. And we discovered quite a bit of variants that were under very strong selections for different things, bacterial, viral, and defense against nematodes and all of that. But this is the point I want you to appreciate, that studying African people benefits non-Africans tremendously, and that is this. There are variants we are calling pathogenic in the US today, in Europe, that a lot of Africans have, and they are running around the streets, and they are not sick. Which brings to question, are those variants actually pathogenic? And if they are pathogenic, do we have other variants in the background that are minimizing the effect of those pathogenicity in other populations, and therefore give us a more robust understanding of the biology? So you can see where we can benefit there. And about, again, in terms of stories, understanding our history, those are, again, very, very important. But this box here, and the box that I showed there, I just want to tell a quick story. The boron, they are a small group in Nigeria, central Nigeria, but they carry about 14% of the ancestry you see in East Africa, in Nilo-speaking people. How did that ancestry go from East Africa to central Nigeria, which is West Africa? We don't know, but again, understanding human history. So I hope I have provided you good evidence in terms of scientific imperative and also in terms of social justice. And again, I'm going to skip this for the sake of time. What I want to, again, message I want to leave with you is that there is diversity in our genome, but the diversity does not rise to the level of self-speciation. Hence, no matter where you or me go in this world today, if we decide we want to share DNA, we will create an offspring. We are completely compatible. Okay? So we are not subspecies. That is a critically important point. Because when I say sometimes that there's human genetic variation, people say, okay, why do you say there's no race? Again, I think that's the important point, and I want to use this cartoon here to just make the point again. And that is, we genotyped a lot of people around the world, and we discovered 21 ancestries. 97% of people in the world are of mixed ancestry, average of 4 ancestries per person. Okay? So when you look at yourself, think about the fact that you are a mosaic of your own history, of your own ancestries, and all of that stuff. And most of us don't have a full appreciation of our ancestral background. We know 1 or 2. But we really do not have a full appreciation for it. And that is why I say that trying to use genetics to define race or define us is like trying to slice soup. You can cut all you want, the soup stays mixed. Okay? And we can make mistakes by looking at people phenotypically. This was an 8-year-old boy that almost went into surgery because he's phenotypically European. Nobody thought about sickle cell. But if you look at the ancestral background, you will see why this kid will have sickle cell disease because of the background that the parents came from. And I asked the audience, where in these countries here, which one do you think should have the highest level of sickle cell disease? Now, because of time, I'm just going to tell you the answer. Most people will say South Africa, but it's not. Has not been sickle cell in South Africa because they didn't have to deal with malaria, whereas Greece and things like that have much higher level. Again, I'm just going to go. So in summary, lack of diversity really hinders our understanding of biology, and we have to do a better job. And I want to invite you to Cape Town in 2023 to join us. We call it Coming Home because, like I said, we are all Africans beneath our skin. So come and join us. Let's understand the genetics of African people. And that is party with my mother and my sisters. And she's absolutely loved by my family. Before my mother passed away, whenever I go to Nigeria and Patty is not there, she would say, "Where is my daughter?" You know. So my sisters up to today, they all cherish Patty, and because she's a fantastic human being. You know, she gives so much and takes so little. So she has made my life a wonderful, wonderful, place. This is her doing field work in Ibadan, Nigeria, our field staff. And then this is her in Kenya, interacting with our colleagues and also the families that we collected data from. Again, so Patty, I want to say what this lecture dedicated to you, there's no way I would have missed it for anything. And I want to really use the opportunity to thank you for all that you have done for me, my family, and for ethics around the world. I challenged Patty yesterday to say, now that you are stepping down a little bit, every day, maybe in the next 1 year or so, just write a page to tell a story. I say you have a story to tell to all these young men and women who want to continue to study ethics in the American context and in the global context. So I hope she takes me up on that challenge. And I want to thank everybody for coming and listening to this. Thank you very much.